Go right ahead.
Thanks, Daniel. So I may not be able to see you on the screen. So if you have any questions during the talk, then do just shout out, and I'd be happy to answer them. And of course, there'll be time at the end for questions. So thanks very much for having me to speak. My name's Paul Roberts. I'm a lecturer in the Department of Optometry and Visual Sciences at City St George's University of London. And I'd like to talk to you today about some work I've been doing for a while now on mathematical modelling of retinal degeneration and metabolism, and thinking particularly about the group of inherited conditions, retinitis pigmentosa. Hopefully you can see my laser pointer here. If you can't, then
It's visible.
say. It's visible, cool. So first, just some brief acknowledgements. So this work's been completed, or the first part of this work was completed in collaboration with Helen Byrne, Eamon Gaffney, Alex Voss, Phil Luther, and Jonathan Whiteley. And over time, the work spanned University of Oxford, Sussex, and Birmingham, and been funded by EBSRC, BBSRC, and through the SMQB. So I thought I'd begin with some definitions just to orientate things in terms of the field in which I work. So I've coined the term mathematical ophthalmology to describe the field which uses mathematical tools to preserve vision by elucidating the mechanisms underpinning ophthalmic health and disease. So using mathematical tools to preserve vision by elucidating the mechanisms underpinning ophthalmic health and disease. And so the sorts of tools I'm talking about here are things like differential equations, models, ordinary or partial differential equations, or stochastic type approaches, but things that are basically getting at describing the underlying mechanisms that determine how the eye functions in health or disease, and then letting the behaviors that you see in the clinic or in experiments emerge from that. And this is complementary to an existing term, computational ophthalmology, which is basically more like artificial intelligence type approaches, where you use computational algorithms to preserve vision by identifying and predicting ocular features and risks. And so in each case, these two fields are complementary in themselves to experimental or clinical approaches, both in three respects. Firstly, in terms of deepening understanding, so you can get a deeper understanding through the use of these approaches than you would through experimental or clinical approaches alone. It enables prediction, so you can make predictions about how the eye would behave in healthy state, in disease, or in response to perturbations. And you can also think about treatments, designing optimum treatment regimens, or perhaps identifying new drug targets, or thinking about how you might even devise personalized treatment strategies. So as I said, these two approaches, mathematical and computational, are complementary to one another, but they do have their own distinctive strengths and weaknesses. So mathematical ophthalmology, as you might have guessed from the terms I've highlighted, gives you a high degree of mechanistic insight. By contrast, computational ophthalmology, or artificial intelligence, gives you low or often no mechanistic insight, for the reason that the algorithms in question don't contain within themselves descriptions of the underlying mechanisms. On the other hand, computational ophthalmology is a very powerful predictor, and can often make predictions just as good or better than the best clinicians, and in a fraction of the time. In contrast, mathematical ophthalmology can make predictions, but that's dependent upon having accurately captured the underlying mechanisms. So as I said, these two approaches are sort of complementary to each other. They each have their own strengths and weaknesses, and there's certainly potential for them to be combined together more in the future, and there have been a few examples of that thus far. And if you want to refer to both terms in one breath, my suggestion is this acronym MACO, mathematical and computational ophthalmology. But I work pretty much within the mathematical side of things, although in time, hopefully, I'll encompass a bit of both. So I just want to give a brief bit of background biology just to orientate us. This is partly, if anyone in the future is listening to this and doesn't know about the biology of the eye, I realise that the people on this talk will know more about this than me, but just to highlight a couple of key facts. So I'm talking here about the retina, which is the light detecting tissue that runs from the optic disc to the ora serrata, and is supplied with oxygen and other nutrients by the choroid, which sits next to it, which also drains away waste products. The main light sensitive cells are the photoreceptors, the rods and cones, and those have a very heterogeneous distribution within the eye and the human eye. So the cones have got a very high density at the center of the retina at 0 degrees eccentricity at the fovea, and then that drops off very rapidly towards the ora serrata, while the rods are absent from the very center of the fovea, from the foveola, and then increase in density quite rapidly up to about 20 degrees eccentricity, and then that drops off more gradually towards the ora serrata. So basically you have a cone-dominated fovea and a rod-dominated rest of the retina. And then both are absent from the optic disc, of course. So in the models that I'll be talking about, I'll be focusing on the outer retina, so the retinal pigment epithelium and the photoreceptors, the rods and cones, ignoring the inner retinal layers. Just to remind you with the rods and cones, so that then you can't, if you've lost them and without some sort of stem cell therapy or something, you can't replace the rods and cones, although the outer segments are repeatedly shed. So the tips of the outer segment, say the top 10th or so of the outer segment of a rod is shed each day and then replenished where it meets the inner segment, a bit less regularly with the cones, but so there's a constant sort of shedding and renewal of those outer segments, and which those tips are then phagocytosed by the overlying RPE. So as I said, the main focus here in terms of the disease is on, or for this talk, is on retinitis pigmentosa, although some of this would be applicable to other conditions, but the focus here is RP. So the term RP refers to a group of genetically mediated retinal degenerative diseases. The mutations in question are typically expressed in rod photoreceptors, sometimes also in the RPE, but not generally in the cones. It would depend a bit perhaps on how you defined RP as to whether you counted examples where cones have mutations. If you're a patient with RP, then you would experience night blindness initially in adolescence, and then as time goes on in early adulthood, you begin to lose peripheral vision, which in time would develop into tunnel vision, and eventually, after a period of decades, say loss of central vision. Although the speed of degeneration varies quite a lot between not just mutations, but even individuals in the same family. It's a leading cause of blindness worldwide, so it affects about 1 in 3.5 to 1 in 4,000 people globally, so about 2 million worldwide. It's currently incurable in the sense that you can't prevent a person from going blind with RP or restore the vision if someone has lost it fully. Although there are treatments available to try and slow degeneration, with which have some sort of limited or various levels of effect. There's a couple of things that so I should note the picture on the right here. These are fundi of healthy and diseased retina, respectively. So this is healthy on the left, RP retina on the right. And the sort of classic things that you notice are there's a sort of a waxy palate of the optic disc. You get attenuation of the retinal capillaries, and then you also get these bone spicule deposits, the pigmentary deposits, from which it gets the pigmentosa part of its name. And that's where RPE cells, so the photoreceptors have degenerated, and RPE cells have migrated from the outer retina into the inner retina, forming little cuffs around the vessels. So there are two particular things that I wanted to note about this condition that are of particular relevance to what follows. The first is that it's a rod cone dystrophy, so rod photoreceptors are affected earlier and more severely than cones, both in terms of their function and also their vitality. And secondly, that there are some quite sort of characteristic patterns of spatiotemporal patterns of degeneration that you see, and hence of visual field loss, where degeneration of the retina initiates in the mid or far periphery in small patches that expand and coalesce, and then that over time spreads inwards. So various mechanisms have been proposed to explain secondary cone loss in terms of the rod cone dystrophy, the first of these 2 points. And I would argue that those mechanisms are also relevant to explaining the second point about these patterns of degeneration that you see, although they're not always necessarily linked together in the literature. And so I'm going to be using mathematical modelling here to try and explore the mechanisms that underpin the secondary cone loss and the patterns of degeneration you see. And I know, sorry, perhaps I should have said the sort of the question here is that, well, the rods express the mutation, so you'd expect them to degenerate. But then why is it that the cones would degenerate afterwards if they don't express the mutant gene? So there's some sort of manner of dependence, direct or indirect, of the cones upon the rods. So there are a number of mechanisms that have been proposed to explain secondary cone loss, and which I would argue could also at least go some way towards explaining these patterns of degeneration. They all start the same way. They start with a mutation that leads to rod degeneration. And then there are 5 main mechanisms that have been hypothesized to explain secondary cone loss, which I'll go through briefly now. The first is oxygen toxicity. So the idea is that you lose photoreceptors, that decreases the demand for oxygen, which then results in an increase in oxygen levels. Those become toxic for the remaining photoreceptors in that area, leading to photoreceptor degeneration, which then of course decreases the demand for oxygen, leading to a further increase in oxygen levels. And so you get this sort of positive feedback or vicious cycle. Second hypothesis is known as the trophic factor hypothesis. The idea here is that the rods produce a trophic factor which is necessary for cone survival, so that if you lose the rods, then the trophic factor is depleted and then the cones degenerate. In this case, cone degeneration will remove the thing that's consuming the trophic factor, so that would then inhibit a further decrease in trophic factor levels in and of itself. There are three other. I'm going to focus on the first two in what follows, but I'll just mention the other three more briefly. So there's the metabolic dysregulation hypothesis, which suggests that there's a disruption in the exchange of nutrients between the RPE and photoreceptors, leading to photoreceptor degeneration. There's a toxic substance hypothesis, which suggests that when rods or photoreceptors degenerate, they release a toxic substance which kills the neighboring cells. And there's the microglia hypothesis, which suggests that when photoreceptors die, that their death triggers the activation of microglia in the inner retina, which then migrate into the outer retina, phagocytose the debris of the dead cells, and then themselves release a toxic substance which kills neighboring cells. So all of these, I know of these mechanisms as self-reinforcing, in that once they've gotten started, they will keep going, with the exception of the trophic factor hypothesis, which is self-limiting in that cone loss will decrease the uptake of trophic factor. And all of them could in fact explain secondary rod and cone degeneration, again with the exception of the trophic factor hypothesis, which would only explain secondary cone degeneration, since it's only the cones that are using the trophic factor in this case. So in my past work, I focused on the first two of these mechanisms, and so I'm going to talk about these in the remainder of the talk, although I've got ongoing sort of interest in the remainder. So the other sort of preliminary to look at, take into account here before talking about the modelling, is the patterns of degeneration, which I highlighted earlier.
I know it's very uncomfortable.
Back in 1998 by Grove et al.
Thank you. You're so soft.
Sorry about that.
Is there a question, sorry? Oh, sorry. So there's a paper back in 1998, Grove et al., where they used Goldman visual field tests to look at the spatiotemporal patterns of visual field loss in patients with RP, and they characterized those into 3 groups and 6 subgroups. And then I've come up with this sort of cartoon diagram to sort of summarize that. So here, the black regions are scotomas, blind spots, and the white regions are regions of preserved vision. And I'll relate this to the diagram on the left in terms of different regions of the retina. So the fovea, para and periphovea, mid-periphery and far periphery, going outwards with the optic disc in the mid-periphery. This is not to scale. So in pattern 1, you get a restriction of the peripheral visual field, which is sometimes accompanied by a para or perifoveal ring scotoma. In pattern 2, you get an initial loss of the superior visual field, which can then wind around inferiorly, either nasally or temporally. In pattern 3, you get an initial mid-peripheral ring scotoma, which can then extend superiorly or inferiorly and then wind around the edge. In each case, you're left with preservation of central vision, though given enough time, that too may eventually be lost. So the approach that I'm going to take then is firstly to use mathematical modelling to isolate a particular disease mechanism. I'll do this first with oxygen toxicity and secondly with cone degeneration. So this is a particular strength of mathematical modelling, that you can isolate a mechanism in a way that might be difficult or perhaps impossible to do experimentally, but it's actually very easy mathematically, because you just choose to include that mechanism and to exclude the others in your equations. So I'll isolate a particular disease mechanism. I'll then look to predict what pattern of degeneration that would give rise to. And I should note, these are patterns of visual field loss, but to relate that to patterns of retinal degeneration, then given the pinhole effect of the pupil, then you're just flipping it vertically and horizontally. So I'll then predict spatiotemporal patterns of disease progression and compare those to what you see at a qualitative level to what you see in patients. And then from that, infer the strengths and weaknesses of the hypothesis. So the hypothesis would be considered to have some strength to it, insofar as it's able to recapitulate patterns of degeneration you see in humans. That would show the theoretical sufficiency of the mechanism, although it wouldn't show that it was the mechanism that gave rise to that pattern. And then perhaps more interestingly, if a mechanism is not able to reproduce a pattern that you see in humans, then that suggests that there must be other mechanisms that are at play in that case to generate that pattern. So I'll begin, first of all, by talking about the oxygen toxicity hypothesis. And just to remind you which hypothesis I'm talking about, I'll put this little oxygen symbol at the top, and then I'll talk more briefly about the trophic factor hypothesis at the end. So just to remind you about the oxygen toxicity hypothesis, the idea is there's a mutation, leads to rod degeneration. As photoreceptors are lost, there's a decrease in consumption, so an increase in oxygen levels. That then leads to oxygen toxicity or hyperoxia, leads to photoreceptor degeneration. Further loss of photoreceptors means to get further increased oxygen levels, and so you get this vicious feedback sort of cycle going on. There are various lines of experimental and clinical evidence to support this hypothesis. So, for example, we know from measurements using oxygen-sensitive microelectrodes in rat and cat models of RP that you do indeed get elevated oxygen levels. We know that in humans, where you can't do those sorts of measurements, that there's an attenuation of the retinal capillaries that I pointed out in the fundus image earlier on, which indicates that there's elevated oxygen levels in the inner retina. You get poor choroidal autoregulation, so even though oxygen levels are high, the choroid is still providing a large supply of oxygen. We know from, for example, pig models of RP that oxidative damage is done to DNA, proteins, and lipids. And on the sort of flip side of that, the efficacy of antioxidants have been demonstrated in slowing degeneration in RP, suggesting that they're neutralizing the reactive oxygen species that result from hyperoxia. So it would seem that there's a strong case to be made that oxygen toxicity is playing a role. It's not the sole player at all. These are complementary hypotheses, but it seems to be playing a role in the retinal degeneration. So I'd like to then to develop a mathematical model and use that to answer, for the purposes of this presentation, 3 questions. The first is, when will a patch of degeneration spread? So I mentioned that degeneration starts in small patches which spread and coalesce. So the first question is, under what conditions, under the oxygen toxicity hypothesis, will a patch remain fixed in its size, and when will it spread and get bigger? The second question is, what determines the speed of propagation of a wave of degeneration? So if it does spread out, what determines how fast that happens? And then last, which patterns of degeneration, which spatiotemporal patterns, are explicable via this mechanism, and then think how that compares to what you would see in a human eye. So I'm not going to go through all the detailed equations here, because you don't really need to know them to understand what I'm doing. So I'm going to explain things more in words. The only equations you'll see, I think, probably almost are the 2 just down here, and those are just for fitting. So to describe the equation more in words, so this is a partial differential equation model, which means that it describes and predicts the way in which concentrations of chemicals or densities of cells evolve through space and over time. A PDE model or partial differential equation model is defined on a particular region of space. In this case, we're in what are called spherical polar coordinates, and I'm looking at the region of space is the region between the fovea and the oris arata, and we're measuring the position by the angle theta, which is a polar angle or eccentricity. So I'm basically depth averaging through the retina, so we're losing that coordinate, and for the time being, I'm assuming symmetry around the z-axis, so we lose the azimuthal direction, the other angle. It's important in this model to incorporate and account for the heterogeneous distributions of rods and cones, and which I mentioned earlier. And so to do this, we make use of some data kindly provided by Christine Curcio from her 1990 paper, where she measured healthy distributions of rods and cones in a number of humans. And so what I do then is I fit mathematical equations to those distributions so that that information can be incorporated into the mathematical model. And so that the top equation here is an expression for the cone density, which is the sum of 2 decaying exponentials, and the bottom equation is for a sort of modified decaying exponential for the rod density. So the fits to the data are shown here. So this panel shows the fit to the rod distribution. So the red curve is my approximation, the black dotted line is the data. And this is from 0 degrees eccentricity at the fovea through to the oris arata. This is rods. Similarly for cones, good match between the 2. And if you add the 2 top panels together, then you get the distribution of rods and cones together, so all the photoreceptors. So what I'm then going to do is I'll incorporate that information into the model. In its simplest form, it consists of 2 governing equations, which I'll describe here in words. So the first equation says that the rate of change of the oxygen concentration, or C, is going to be dependent upon the diffusion of oxygen, so the way it moves towards the oris arata or towards the fovea. Its uptake by photoreceptors, which will happen in a manner which is dependent on the concentration of oxygen and the density of photoreceptors at a particular location. And then also an exchange of oxygen with the choroid, which in general is an input term because the oxygen concentration is going to be, of course, higher in the choroid than the retina. The second equation then is for the rate of change of photoreceptor density through space and over time, and that's given by P. And basically, the photoreceptor density will remain fixed unless and until the oxygen concentration exceeds at a particular location the hyperoxic threshold, which I denote as C crit. And when that happens, then the photoreceptors in that region will begin to decay exponentially in their density. In order to close this system, we need mathematically two other things. We need, first of all, some initial conditions. We need to say what state the system is in at time t equals 0. And for the purposes of this presentation, we're always going to start with an initial condition where I remove all of the photoreceptors from some interval θ1, θ2. So in one dimension, this would look like this then. So between θ1 and θ2, I've removed all the photoreceptors. So this red curve goes down to 0 between 0.4 and 0.6. And as a consequence, the oxygen distribution, the black curve, comes up to sort of choroidal or near choroidal levels in that interval. Yeah?
I just wanted to ask real quick, is oxygen consumption by rods and oxygen consumption by cones being treated equally in this model?
For the purposes of this model, it's being treated equally.
Okay.
But you could easily modify it so as to have different levels of oxygen consumption by each.
Yeah, thank you.
I don't think it would. I think if you altered it, it wouldn't have a particularly large effect on the actual results. It would be a sort of the qualitative behavior would be the same. There'd be slight shifts in the quantitative effect.
Sure.
That's a good question. And then in terms of the final condition you need are boundary conditions. And then what we say is that oxygen can't escape through the boundaries. So that occurs at the fovea just by symmetry of the problem. And at the ora serrata, it's basically the simplest kind of assumption that we can place. It's a sort of reasonable assumption. And you could choose other boundary conditions there, but ultimately it wouldn't have very much of, for any reasonable boundary condition, it wouldn't have much of an effect on the results. Ultimately, they'd be confined to that sort of edge region. So to summarize then, the inputs to this model are θ1, θ2, and C crit, so the edges of this boundary of loss of the initial condition and the hyperoxic threshold, and the output to the evolving concentrations of oxygen and density of photoreceptors through space and over time. So I should say it's not a biochemically detailed model. That's something I'm looking to go into more detail in sort of current and future work, but this is sort of it is a mechanistic model, but it's also a somewhat phenomenological level in that it's sort of agnostic to some of the finer details of what's going on, but would be consistent with them. So if I go through then each of these, come back to the questions that I posed earlier on now that we've formulated the model. The first was, when will a patch of degeneration spread?
Real quick.
So to answer, sure, yeah.
Can I also ask, where does C crit come from in the model? What's the value, or where did you derive it?
Sure, that's a good question. So C crit is chosen to, well, the highest it could be would be the level of oxygen in the choroid. So that's an obviously upper bound on C crit. The lower bound on C crit is the level at which so if you put it below that level, such that even for a healthy distribution of photoreceptors, it would start to degenerate even under healthy conditions. So it needs to be high enough that a healthy retina remains healthy, but low enough that it's not at choroidal levels. So that gives you a sort of a window in which it can sit. And then once it's within that window, again, the qualitative behavior of the system is roughly the same, and you can characterize how variation of C crit would influence that. But basically, it doesn't really matter too much what the value is within that window in terms of what I'm going to tell you today. So if we're looking at degenerate patch stability and whether or not these patches will remain fixed in size or expand in terms of the oxygen toxicity hypothesis, then it's helpful to go back to this diagram I showed you earlier, where we've removed a patch of photoreceptors between θ1 and θ2. So if the oxygen concentration at this right-hand boundary was above the oxygen toxicity threshold, that would mean that there would be some region to the right of the boundary where the oxygen concentration was still above that hyperoxic threshold, but yet in which photoreceptors were currently healthy. And under that circumstance, those photoreceptors would then be caused to degenerate, and as a result of which the patch would expand outwards to the right. So that would be the condition for the patch to expand at that region. If the oxygen concentration, though, were at or below the hyperoxic threshold, the oxygen concentration outside of the patch would also be below the hyperoxic threshold, and therefore it would remain stable. So what it's going to depend upon is the oxygen concentration at each edge of the patch. And so here we're looking at a steady state problem. So it's one that's not evolving through time. So we're simplifying the maths a bit, and then we'll come back to a dynamic problem that changes in time in a moment. So there's basically 4 scenarios you can have then. Either it's the case that the oxygen concentration is below the hyperoxic threshold at both boundaries, and then the patch will remain fixed, or it could be above on the right and expand out peripherally, or on the left and expand centrally, or on the left and the right and expand in both directions. And one way to map this out is with this diagram on the right. If you don't understand the diagram on the right, don't worry about it. You don't need to understand it to understand the talk. But for those who find this sort of way of expressing it helpful, I thought I'd put it in. So here, this is dividing up θ1, θ2 parameter space. It's upper triangular because θ2 is always to the right of θ1. Regions of type A that are in green are stable, so they won't. So if the patch is at that position and width, then it won't expand. Regions of type B will degenerate leftwards or centrally in orange. The light blue ones of type C will expand rightwards or peripherally, and the regions of type D should expand both centrally and peripherally in red. Another way of expressing this is to say that you can divide the retina up into a series of 5 regions which alternate between being unstable and stable. So there are 3 unstable regions and 2 stable. So relating this again to the eccentricity between the fovea and the oris arata, what you get is a peripheral unstable region, a mid-peripheral stable region, a para or parafoveal unstable region, a near-central stable region, and then really close to the origin, a central unstable region. And to be clear what I mean by stable unstable region here, if the boundary of a patch of degeneration lies within an unstable region, then it will expand outwards until it either reaches a stable region or the edge of the retina. Whereas if a component of a boundary lies in a stable region, it will just remain fixed in space. Another way of expressing this diagram is to show it kind of a bit like the fundus image I showed before, sort of on FAS. So what you get is a sort of a bullseye pattern of alternating unstable, stable, unstable, stable, unstable regions arranged roughly like this, but arranged like this. And if you were to change that secret value, which Daniel asked about before, then the effect would, you still get this sort of bullseye pattern, but you just move where the interfaces were between the stable and the unstable regions. So that was looking at what's called a steady state problem, one that doesn't change in time, which involved some simplification and mathematical analysis under the bonnet. But then what's nice we can then do is look at the full dynamic problem, the problem that changes through time, and see if that full problem, mathematical problem, matches up with the simplified version. So I'm now going to show you some simulations of the dynamic problem that evolves through time, and I'll do that with some heat maps. So there's an example here, for example, which goes from the fovea to the oris arata again, the increasing eccentricity on the x-axis, time increasing upwards. And here, a red or hot sort of colour means you've got a high density of photoreceptors, and a blue colour means you've got a low density or no photoreceptors. So here we take out a great big patch and it expands outwards to the right, peripherally, and inwards towards the centre. Whereas in this example to the left is a narrower patch, sort of somewhere in the mid-periphery, say, and here it doesn't expand at all. The 2 interfaces are just vertical and straight. So what I'm going to do is I'm going to take out patches of photoreceptors from it so that they're within these 4 different regions where it either remains fixed or goes left or right or both, and see if we get the same kind of pattern. So let's say I'll take a patch from region A where it should remain fixed. Then we get the behavior we expect, the photoreceptors remain fixed, and I also just plot for completeness the corresponding oxygen concentration, which is high where the photoreceptor density is low. Or I could take a region in patch in region B where it should go inwards but not outwards. And again, that's the sort of behavior we get. So it goes inwards towards the fovea but not outwards towards the oris arata. Or a region of type C where it should go outwards but not inwards. Again, it does the right kind of thing. So it's going out towards the periphery but not inwards. And then lastly, a region of type D where it should go both outwards and inwards. And again, that's what happens. So this answers then the question of when will a patch of degeneration expand or remain stable? The answer is it depends upon where the boundaries of that patch lie in relation to these stable and unstable regions. But this leads us then on to the second question, which is the speed of propagation. And you'll note that this isn't in general constant, it varies. So for example, in this plot here, you'll see that the wave of degeneration accelerates as it goes towards the periphery, because this isn't just a straight line, it's a contour that's bending around to the right, so that means it's accelerating. And similarly on the left, it accelerates initially and then sort of slows down as it moves towards the centre of the retina. So then the second question was, what determines the speed of propagation of a wave of degeneration? And I think this is the only other equation I was going to show you. And basically, you can do some mathematical analysis, and you can actually derive a mathematical expression for the wave speed of degeneration, v, in terms of parameters which are known in your model. So d here is the diffusivity of oxygen, beta is the exchange of oxygen with the choroid, secret is that hyperoxytic threshold, and q is to do with the rate of oxygen uptake by photoreceptors, and the p tilde is the local density of photoreceptors. So this is a decreasing function of the local photoreceptor density. So it means that as the photoreceptor density decreases, the wave speed increases, which is what you would expect in that if you've got a lower local density of photoreceptors, that means that the oxygen concentration is going to be higher, and so you would get a larger hyperoxic region ahead of the wave of degeneration, meaning it's going to expand more rapidly. And so if you were to plot v as a function of theta then, because p tilde here is a function of theta, the eccentricity, you'd get a plot that looks something like this. So you'll see this sort of matches up with the simulation I showed you a moment ago, in that the speed increases as you move towards the ora serrata. So that was the sort of that bending contour as it bent around to the right. Or similarly, it sort of increases in speed as you move towards the fovea, and then decreases again and again. We saw that pattern of change in the wave speed. And so the other useful thing about this expression is it might sort of look, if you're not a mathematician, it may look a bit complicated, but actually you can easily just sort of stick this as a formula into Excel and very quickly calculate the value of v based on these parameters. Whereas if you wanted to, so you'd like to do it in a fraction of a second in terms of the calculation. Whereas actually running a full numerical simulation takes more on the order of hours or something. So it sort of speeds things up quite a lot. And then finally, the third question to look at here is which patterns of degeneration are explicable via this mechanism. And so I'll just remind you, these are the patterns of degeneration that you see in RP, or in terms of visual field loss, but which, if you flip vertically and horizontally, correspond to patterns of retinal degeneration. So here I'm going to go from a 1D model that I had before to a two-dimensional model. So now, in addition to considering the polar angle theta from 0 to 1, which incidentally I scaled, so this wasn't degrees or radians, this is a scaled value. I'm also going to take into account the azimuthal dimension, so phi, which is how it's spinning around the z-axis. So let's say I were to look at the healthy distribution of photoreceptors, it would look like this if you paint them over a sort of a hemisphere in terms of their density. So you've got a high density at the center of the fovea there in red, and it drops in blue and then goes up again and so on. And so that you can see everything in the simulations I'm going to show you, what I'll do is I'll project that onto a 2D plane like this, and then flip that up so it's on fast, so that you aren't having bits of it hidden behind the hemisphere. So what I'm then going to do is I'm going to sort of damage the retina in a couple of locations. I'm not going to remove the photoreceptors because, of course, they can't grow back if you've removed them. The idea I've just sort of damaged their vitality in some way. So in this case, I've damaged some photoreceptors in the mid-periphery, and here they recover. So the mid-periphery is pretty robust to damage under the oxygen toxicity hypothesis. Whereas if I do the same thing, but I move the damage closer to the periphery, a different phenomenon occurs. So here, time is marching onwards, left to right, 1, 2, 3, and then 4, 5, 6 at the bottom. So what you find now is that this patch consolidates, expands outwards to the edge of the retina and winds around the periphery. And then these two arms keep going around the edge until they meet at the far side. So this actually replicates the mid-stage of pattern 3 that I mentioned earlier. Another scenario that we could consider is we could just start with healthy initial conditions, but introduce another kind of degeneration. So up until this point, I've said that all the degeneration that occurs is due to oxygen toxicity. But of course, the rods are also degenerating simply just due to a mutation independently of that. So you could include that kind of degeneration as well and see what kind of patterns of degeneration that leads to, given the knock-on oxygen toxicity degeneration that will result. So here I'm just plotting the cone distribution. I'm taking the log just to make it easier to visualize. Again, time is increasing 1, 2, 3, 4, 5, 6. And what you find is now that you get degeneration at the periphery of the retina, expanding inwards, and then also a parallel peripheral region that comes outwards from the fovea and sort of meets somewhere in the mid-periphery. At the end of this, you may not be able to see it. I'll put my laser pointer there. There is a red dot somewhere in the middle there, which is the preservation of the fovea. So this replicates then pattern 1b that I mentioned earlier on. So to summarize all of this then, these are the patterns of degeneration you see in vivo, and these are the ones that you see in silico from the model. So we've replicated pattern 1b and also the last stage of pattern 3. but we're not able to, which is the winding around the edge. But what we're not able to reproduce are cases where there's a preferential loss of the mid-periphery, so say the early stage of pattern 3, or just general loss of superior or mid-periphery of the retina in pattern 2. So to summarize this first much larger part of the talk then, these are the first models that consider the mathematical models to consider the oxygen toxicity hypothesis, or to account for the distribution of photoreceptors. We've determined the topographical regions in which a patch of photoreceptor loss will spread or remain stationary based on those stable and unstable regions that I talked about. We've predicted that the wave speed of degeneration as these patches expand and coalesce is a decreasing function of the local photoreceptor density, which can be precisely quantified, and we're able to predict the spatiotemporal patterns of retinal degeneration, and therefore to demonstrate the strengths and limitations of the oxygen toxicity hypothesis. That is to say, we've shown that it's sufficient to reproduce some, at least qualitatively, some of the patterns that we see in humans, but there are other patterns that it is theoretically unable to reproduce, that so suggest that other mechanisms must be producing those patterns. So I'll move on now much more briefly just to talk about the trophic factor hypothesis. So just to remind you how this works, the idea is that there's a mutation, leads to rod degeneration. That then leads to a decrease in trophic factor levels because the rods are producing the trophic factor. That then leads to cone degeneration because they're depending upon the trophic factor for survival. But then as they go, then they're not consuming trophic factor so much. There's a limit placed on the amount by which the trophic factors decrease. So there's quite a bit of experimental evidence from this. Arguably, it's perhaps the most studied of the mechanisms so far. So the trophic factor has been identified as RDCBF, or rod-derived cone viability factor, and this was identified by Thierry Levard et al. Back in 2004. And so it's known as this kind of protein that's produced by rods. And its mode of action has been identified. So this was back in 2015. So they know that it binds to basogen 1, which in turn binds to GLUT1, which then increases the rate at which cones uptake oxygen. And it's also been shown to slow cone degeneration and preserve cone functions in chick, rat, and mouse models of RP. So there's pretty solid evidence that, again, that this trophic factor RDCBF is playing an important role in RP. So I'm just going to look at, to keep the presentation to time, I'm just going to look at one modelling question here, which is which patterns of cone degeneration are explicable via this mechanism under the simplest of assumptions. And note that I'm only talking about cone degeneration now, since this mechanism only by itself explains the loss of cones, not rods and cones. So I'm returning again to this sort of model, a 1D model, so it's between the fovea and the oris arata. And again, I'm incorporating rods and cone distributions as before. Going to the governing equations, so in its simplest form, we now have 3 governing equations. So instead of the oxygen equation, we now have one for the rate of change of trophic factor concentration, F, and that's due to the diffusion of trophic factor, so moving towards the oris arata or towards the fovea. It's produced now by rods rather than coming in through the choroids with the oxygen. We're not looking at treatment here, but it could be input via treatment. It's taken up by cones, and there'll be some sort of biological decay. And now we're distinguishing between rods and cones, so that the rate of change of rod density, denoted by PR, will either be such that there's the density of rods may remain fixed, or it's decaying exponentially to replicate mutation-induced rod loss. And then in terms of the cone density, we'll either say that that's fixed, which is PC, sorry, that will remain fixed unless and until the concentration of trophic factor, which could be RDCBF or in theory some other trophic factor, it could be the case. So it'll be the case that the cones will remain healthy until the local concentration of F drops below the critical threshold F crit, at which point they'll just decay. So before we had a situation where if oxygen levels went too high, then photoreceptors degenerated, whereas now we've got a case where if the trophic factor concentration goes too low, then cones will degenerate. And then as before, we need initial and boundary conditions. So in this case, we're just going to look at healthy initial conditions. So all the cones and rods are there healthily and happy. And for the boundary conditions, it's the same as before in that we're assuming there's no flux of trophic factor out the boundaries. So that's by symmetry at the oris arata and at the fovea, and the simplest assumption at the oris arata. And so now the inputs, the rate of mutation-induced rod loss and this trophic factor threshold, and the outputs, the evolving concentration of F and densities of rods and cones through space and over time. So the only question I'm going to look at here is just which patterns of cone degeneration are explicable via this mechanism. And the answer is given in this panel down at the bottom left. So here I'm plotting the proportional number of cones remaining at a given point in space, at a given point in time. So it's the if it's yellow, it means they're all there. And if it's blue, it means they've all been lost. So I'm just dividing at each location the current density of cones by the healthy density at that location. So here again, we're going from the fovea to the ora serrata, time is marching upwards. So here what happens is degeneration starts at the center of the fovea and then expands outwards towards the ora serrata over time. So this is both surprising and unsurprising. It's surprising in the sense that perhaps, as I said, this mechanism has received the most attention, and yet under the simplest of assumptions, it's not reproducing the pattern that you would see in RP. So in RP, it starts somewhere at the periphery, mid-periphery, and expands inwards, whereas here it's starting right at the very center and expanding outwards. In some ways, that's not surprising, though, of course, because if you think about the ratio of rods to cones, then it's a monotone increasing function. It's lowest at the fovea, and it increases as you go out towards the ora serrata. And so this is based on that ratio. It's precisely, under the simplest of assumptions, the pattern that you would expect to see. The degeneration would start at the center and move to the edge. So to summarize this section, then, the trophic factor hypothesis cannot, under the simplest of assumptions, replicate the in vivo patterns of degeneration that you see in humans. I haven't got time to talk about it here, but I have then gone on to look at this hypothesis in a bit more detail and to think, would there be some conditions under more complex assumptions under which you could reproduce those patterns of degeneration? And there, what you can do is you can vary the spatially the rate of mutation-induced rod loss, which is known to vary spatially in humans, or you can look at varying the threshold, the critical threshold for trophic factor concentration. Which again may well vary spatially. So there's reason we know that, for example, there's a heterogeneous expression of other trophic factors which confer, additional survival or robustness to certain parts of the retina. So there's reason to suppose that that F-crit may not be uniform. So we can basically solve what's known as an inverse problem, and say, for a given pattern of degeneration, how would they have to vary to produce that? And so in theory, there's some experimentally testable conditions there as to how you might. What would have to be the case in order for the trophic factor hypothesis to produce those patterns. So where was this going in future then? So far I've looked at these two mechanisms, oxygen toxicity and trophic factor depletion. And I'm sort of beginning to look at these other things now, metabolic dysregulation and toxic substance or microglia hypotheses. And you can look at these each in isolation. And some of these will involve much more detailed sort of biochemical modeling. But then you can also think about how those might then, once you've got models for each of these things, how they might combine together to give the full range of patterns that you see. So, for example, pattern 1 could be a combination of oxygen toxicity and trophic factor depletion. Pattern 2 might combine all 4 of these mechanisms, and pattern 3 again could combine multiple mechanisms. Now, it might be the case that there's more. I'm sure there are more ways that you could put these mechanisms together to maybe produce these patterns. But the important thing here is that if you're a clinician and you're treating the condition, then the results are sort of indicating that there may be different temporal phases of the disease during which different mechanisms are dominating in terms of what's causing degeneration. And it may also be the case that depending on the spatial location in the retina, again, different mechanisms might be dominant in driving further degeneration. So that would have implications for how you would then treat. But before I close, I just wanted to highlight a couple of things in case they're of interest. So submissions are now closed, but if you'd like to learn more about the wonderful world of mathematical and computational ophthalmology, I have a special issue in IOVS. There's still a couple more papers to come in, but I think there's about 38 or so up there. So do take a look, and that includes invited reviews from the following people. And so there's a QR code there if you want to find it, and a link. Oops, sorry. If I might be really cheeky and just advertise another complimentary seminar series that I've run, which is on mathematical and computational ophthalmology. Again, the link is here. So this is much more irregular than Daniel's, so not nearly so well organised. But it's so the talks we've had so far are there on the right, and hopefully there'll be some more in coming years. So do look out for that. And then lastly, if you happen to be here and you're an early to mid-career researcher, or know any early to mid-career researchers, then you might be interested in this paper that I published last year called Advice to a Young Mathematical Biologist. It's not just for a mathematical biologist, it's really would work for anyone in the experimental, clinical, biological sciences, physical sciences. The advice doesn't come from me. It comes from 10 current and former presidents of the Society for Mathematical Biology, and covers topics like deciding if a career in academia is right for you, finding and working with a mentor, formulating a research question, networking, applying for faculty positions. So I found the advice in there quite useful, and hopefully you or other students might find it useful too. So in case that's of interest, I've again just put the link up there. So I'll close here then, just to say I've been talking about inherited retinal disease today, but I've ongoing interests in that. I also work in retinal metabolism, oxygenation, AMD and its treatment, and also visual neuroscience, and a few things outside of that, including microbiology and a bit of cancer stuff. My main focus is very much on the eye. If you wanted to contact me, my email address is there. I've got a web page if you want to find a bit more on a Twitter handle. So thanks ever so much for listening, and I'd be happy to take any questions that you might have.
All right, thank you very much. Do we have any questions?
Can I ask a rather naive, perhaps silly question? At the beginning, you mentioned that the capillaries were declining in the RP condition, and that was associated with increased oxygen. And I'm having a hard time kind of understanding that. Am I missing something? I thought the capillaries delivered more oxygen.
Sorry, what I meant was they're reacting to the fact that oxygen levels have risen, so that they're constricting so that they maintain the levels at normal. So actually, I think if you measured the levels in the inner retina, they'd be normal, but it's because they've been constricted to compensate for the fact that were they at their normal level of dilation, then it would result in hyperoxia or elevated oxygen levels, at least in that region. So it's showing that there's increased oxygen coming from the outer retina because there's decreased consumption there.
Got it. So the capillaries are receding to balance the oxygen in the retina. Okay.
Yes.
Thank you.
That's right.
So I have a question. So it was nice to talk, Paul. Thank you. None of the retina is really stable in RP because degeneration proceeds to completion. And so I'm a little confused about the idea of a stable region. So what that means is that the oxygen levels and the consumption of that region never managed to exceed the critical value. Is that right?
Yeah, so it means that as far as oxygen toxicity goes, the density of photoreceptors is high enough that even if you lost a patch of photoreceptors in that region, if the boundary of the patch of loss lay within that region, it wouldn't expand out due to oxygen toxicity alone. You would need something else to cause the patch to expand. So oxygen toxicity and those assumptions wouldn't be enough to cause an expansion of the patch, because the photoreceptor density is high enough there that it's able to prevent a hyperoxic region from emerging in a healthy region of the retina neighboring the patch of loss.
Right. So essentially the critical value is never exceeded.
Yes. Yeah.
Because of the balance. Right.
Yes.
Yeah. So the other issue is why does a particular patch start the degeneration? Have you come to grips with that at all?
So, I mean, it's not, as far as the oxygen toxicity mechanism went, if I started with a patch of loss, then that was just an initial condition. So it wasn't seeking to explain that. It was more seeking to say, well, why does it expand or not after that point? If you had mutation-induced rod loss, which I showed at one point, which gave rise to that paraveal ring scotoma, or loss and the restriction of the degeneration from the periphery, for pattern 1B. So if you had mutation-induced rod loss, then that can, because of the distributions of rods and cones, that will give rise to consequent cone loss, because the loss of rods will lead to an increase in oxygen levels, which then leads to So that would, but that doesn't really give you patches as such. That just gives you a more larger regions of loss. Under oxygen toxicity mechanism, it's not clear to me that particularly explains those initial patches. I think perhaps for that sort of initial patchy loss, the best sort of mechanisms, at least thinking about it from a more mathematical perspective, might be something like the toxic substance or microglia hypotheses, because then you just have all it takes is just a stochastic sort of random loss of a photoreceptor somewhere. Then if that results in the release of a toxic substance or the microglia cell coming down and releasing something toxic, that could then amplify up that small sort of stochastic effect and result in a much larger region of loss. So that seems like a better bet for the initial formation of a patch in terms of those mechanisms. Whereas then something like oxygen toxicity could then take it from there and expand it further.
Yeah. Okay.
Okay. If I may.
Francois, Steve's hand was raised for a little bit.
Oh, okay.
First.
Let's see that.
Hi, Rob. I enjoy your talk. So you said the most common pattern is actually the mid-periphery, like an arc loss, and if oxygen doesn't account for that kind of pattern, so what would the other listing that you showed in the beginning could account for this kind of mid-periphery rod loss, which is the most common seen in people?
So I haven't modelled it in detail yet, but I'd say the most likely bet is either the toxic substance or microglia hypotheses, in that if you've got a higher density of rods in that region, you're more likely to get, probabilistically speaking, the loss of rods there, which could then be amplified up and lead to the loss of photoreceptors in those regions. So if the probability of losing a rod in any given location of the retina, naively at least you could say, would be proportional to the local density of rods. So where the density is higher, there's a larger chance that one's going to be lost just stochastically. And once one is lost, then if it releases a toxic substance or leads to microglia activation, they release a toxic substance, then that could then amplify that up in that region. So you're more likely to get an earlier formation of loss of degenerate patches in the mid-periphery under those hypotheses, I would say.
Microglia activation will go with the density of the rods, and so typically the superior arc is more common, except rhodopsin patient. Rhodopsin patients tend to have inferior loss. So the reason you see a superior arc where it starts is because of the density of microglia that goes with the density of rods.
Okay.
I'm asking, is that why?
Well, I'm hypothesizing that it could be the case that this is why the higher density of rods means that's more likely to occur. I haven't demonstrated it mathematically, although my intuition is that that's precisely what the model would do based on sort of thought experiments.
All right, Francois.
Yeah, I would have a question concerning this trophic factor or simulations of this trophic factor idea. Mm-hmm. So, if I understood you correctly, you said that the release of the trophic factor should be proportional to the rod density and the relationship of rod to cones, and you would hence expect a degeneration progressing from the center towards the periphery, from the center where you have lowest rod density and then towards the periphery where the rod density is highest.
Yes. Under the simplest of assumptions.
You said that there might be more complex assumptions that might, I don't know, still reconcile the degeneration pattern seen in patients with the trophic factor hypothesis. Could you expand a little bit on this? What kind of assumptions would you have to make the model fit the data, or
sure,
the way around, the data fit the model?
Sure, no, that's a good question. So for the full answer to that, I've got a paper in Frontiers in Aging Neuroscience or something from 2022, which addresses this. But basically, there are a number of different parameters in the model. most of those you would expect to remain fixed in space. So they should be the same whatever the spatial location. But there are two parameters, and just two, that you might expect to vary spatially. And those are the rate of mutation-induced rod loss and the critical trophic factor threshold, below which, if the concentration of RDCVF or whatever the trophic factor might be drops below that, then the cones would be lost. So both of those could be thought to vary spatially. So I think we know already from measurements of loss of rods in humans, I think this is from again Christine Curcio's data, that that does vary spatially. So there's good reason to suppose that the rate of rod loss varies spatially. But there's also, I think, reason to suppose that it's likely that the critical trophic factor threshold would vary spatially, that there might be parts of the retina that are protected against loss, such that even if the amount of RDCVF or trophic factor is lower in certain regions, there are other things that are there to protect the retina against that. So, for example, I can't remember quite which trophic factor this is, but there are other trophic factors which are expressed not heterogeneously across the eye, and which would therefore provide greater protection in some regions than others, which might perhaps compensate for the fact that RDCVF is not being produced uniformly across the eye. So either of those two could vary, I would argue, spatially, the rate of mutation-induced rod loss or this critical threshold. And if that's the case, then you can do what's called an inverse problem, where you start. So the forward problem is, I know what the parameters are, I solve my model, and then I get out a pattern of degeneration at the end. The inverse problem is you go the other way. So you say, I know what pattern of degeneration I want to get, therefore what has to be true about my parameters in order to get that pattern of degeneration. So you could infer what the spatial profile would have to be for the rate of mutation-induced rod loss or for the critical trophic factor threshold in order to produce that pattern of degeneration. So that's what I do in that paper. You can't infer both simultaneously. You'd have to keep one fixed and infer the other, because there's too many degrees of freedom otherwise. But assuming one was fixed and the other varied spatially, you could you can work that out. Or indeed, if you knew in advance what one of those was, you could work it out exactly.
Okay, thank you.
That's right.
Rob?
Right. I'm just wondering, what would it take to non-dimensionalize the time? Well, you non-dimensionalize the time. What would it take to put it back into the time space so that you could actually see whether the rates you predict match reality?
It's very simple to put it back into time space, in that it's just multiplying that axis by a certain factor. I can't remember what it is off the top of my head, but that's easy to do. I think there is, if you were to do that, then the rate of degeneration from the parameters that I use is consistent with what you would see in patients. So it's the degeneration occurs over a period of decades. I think there's a certain amount of flexibility in. Oh, it's a while since I did this now, but I think maybe there was one parameter where you had a certain amount of freedom. It might have been the secret, where you could basically choose it so as to get a match with that sort of patch rate of degeneration. But most of the parameters are simply just fitted from or taken from the literature. But I think there may have been one that there was a bit of freedom, but maybe I didn't have to tune it at all. Maybe it just did it by itself. But the answer is it degenerates over the right time scale over a period of decades.
Jim?
Yeah, it's just a kind of general question. Do you think the oxygen toxicity mechanism is relevant for AMD also? If the photoreceptors aren't getting enough fuel, for example, they wouldn't be consuming as much oxygen, and oxygen might build up. And if that happens, you would probably get a totally different spatial distribution of degeneration. Have you thought about that?
Yes, I mean, if in many ways the oxygen toxicity stuff that I showed you wasn't specific to RP. I was comparing it to patterns in RP. But if you get a situation where you've lost some photoreceptors from a given region, or where there's been a loss of some cells, or there's a decrease in uptake, so that the oxygen levels have risen, then you'll get the same kind of phenomena occurring. So yes, you would expect to see oxygen toxicity-driven loss in AMD and probably a range of other retinal diseases as well.
So
one other thing I was thinking about. So in in those old Christine Curcio papers that you cited, I think she not only mapped the distribution of the rods and the cones, but she also mapped the sizes and distributions of mitochondria too, which would be the oxygen consumers. I could dig out those references, but I think they were in the papers that you cited.
I think so, yes. I'd have to go back and look, but I think so, yeah.
I should know the answer to this question, but is the rate of spreading of geographic atrophy in AMD similar to the rate of spreading of the degenerative region in RP? If theoretically they could both be a result of oxygen toxicity?
That's a good question. Off the top of my head, I'm not sure. But if there's anyone here who knows more about that, what the rates, how they would compare.
This is Jason. The rates for geographic atrophy are highly dependent upon the position in the from the foveal center. So there isn't a uniform, of course, rate. It always expands at a faster rate the further away from the fovea until you reach the until you reach sort of right around the arcades, and then the expansion rate slows down again past the arcades. Just always been a mystery about why that's the case, that the expansion rates are so topographically dependent.
Yeah, so I mean that seems like it could work with the mechanism that you're speaking of, Paul.
Yes, yes. So you do see that sort of, I showed you in that graph, there's sort of a hump of increase and decrease in speeds that could be consistent then, yeah.
And is there any, oh, so Scott, please go ahead.
Thanks. In the Curcio papers, you have the averages for the densities. Do you know how the beta values that you use could be constrained to reproduce the variability in rod and cone density? And then if you took a theta 1, theta 2 patch, but instead of like changing that, you varied the natural density of rods and cones, could you get even different rates of expansion because now you've shifted those density ratios? Have you tried anything like that?
Good question. I haven't. I think I probably have played around with different parameter values, which would be equivalent to doing that. So basically, the result would be you would change the positions of the boundaries between the stable and unstable regions, and you would change the speed at which propagation expansion occurred. And you can kind of see that. I haven't taken it away now, but there was an equation I put up for the speed of propagation, and it was in terms of, it included terms that it was basically the p tilde term that contained within it the expressions for the distributions of rods and cones. So that was a monotone decreasing function of the photoreceptor density. So if you had a different patient, particular patient, or varied it, so that you had a higher or lower distribution density, then that would have a corresponding effect on the speed. If you decrease the density, or increase the speed if you increase the density or decrease the speed, or perhaps stall it entirely if it's moved into that region into one of those stable regions. That's a good idea.
Thanks.
I haven't tried it explicitly as you've described, but that would be the effect.
Okay.
Okay, I have not to obsess about secret, but to me it seems critical. So it's also, I guess the most important point for me is that it seems experimentally determinable. That
Yes.
you would know what level of oxygen it is enough to kill a photoreceptor. And I guess I'm surprised that there wasn't some literature value that you could easily, find, but that, there's always holes. So,
yeah, I think it's possible, since I was doing the work, that the people have come up with that. At the time, I couldn't find anything, and if people know of a value, then please do let me know. I know that at the time it was known that under hyperoxia, it takes about 3 hours for a photoreceptor to die, and I think that actually that's partly how we got. So Rob was asking earlier about whether the rate of degeneration is correct. I think I've chosen the parameter to be consistent with that, so that it would be a sort of a 3 hour for rod to die or photoreceptor to die, and then that gives you the right speed of degeneration effectively.
Yeah. Well, I guess my question is trying to factor that in with mutation dependent. So is the idea that I have a mutation in rhodopsin that causes gradual stochastic rod death. And, I have a region, two rods happen to have died near each other, and suddenly there's more oxygen there, as opposed to, randomly one over here, one over here. So that's kind of a small patch size, I guess, and would require that secret be, I guess, a little lower, right, in order to make that patch size then cause a spreading event. Have you just sort of determined what patch size you need in order to cause a spreading event under different levels of secret? Does that make sense?
Yes, that makes sense. So for a given level of secret, I can tell you what the patch width would need to be at any given position across the retina, and then you can just within the equations you can just vary the parameter which is secret, and that'll tell you how that minimal width varies from region to region. So yeah, it's sort of I haven't got the diagram here, but if the there's a sort of an interesting thing that happens mathematically when the patch gets very narrow, in that so where you have a patch of photoreceptor loss, the oxygen concentration at the edge of that patch actually varies quite rapidly. And this is because the rate of uptake and supply of oxygen dominates over diffusion. So that means you have a very sharp interface. If diffusion dominated, it would become a lot flatter. But because uptake and supply dominate, it's quite a sharp interface, so diffusion can't smooth that out so much. When you're doing a certain kind of a mathematical analysis, you get these sort of boundary layers either sides of these interfaces. Sorry, my camera's going a bit strange. Let me switch over to the other one, and then I won't flicker so much. So when you have a very narrow patch, so these sort of interfaces overlap each other, and so regardless of the location of the patch, if there's two sides of the patch are close enough, you can sort of get away without the patch expanding if it's sufficiently narrow. But it has to be increasingly narrow as you move towards either the fovea or the orbis serrata, and can get away with being wider if it's more towards the mid-periphery. But I can't, off the top of my head, I can't remember what the values are for the minimum width. But it's in theory the answers are all there in the algebra of the paper. You just need to stick in the right numbers to find the answer for any particular parameter combination that you like.
Yeah, I mean, I guess I'm trying to wonder whether or not clinical imaging data could give you an answer about secret. But
yeah,
maybe not. I mean, maybe they don't catch it early enough, Okay.
Sure.
Yeah, so it seems like everyone's leaving because we're a little bit over time, but we're almost always a little bit over time. So I really appreciate you giving this seminar. I thought it was really great. And thanks for taking the time, Rob. Or Paul, sorry.
That's all right. It's a pleasure.
I was right next to you on my screen.
Thanks very much. It's been a pleasure. And thanks for the questions. They're really, really good questions.
All right. Yeah. All right.
Thank you.
Thanks, everyone.